Hi everyone, and thank you for viewing my chat. My name is Ben Schultz. I'm a current PhD student at the University of Guelph, and I'm going to be talking about a paper that I've recently published that came out of my master's thesis that I wrote along with Dr. Janet Koprivnikar, who was my master's supervisor. Generally, when we think about food webs, we don't include parasitic taxa within them, unless we're talking about parasitic taxa and their impacts on their host species, in which case they're included, but really only through the change dynamics of the host species itself after being parasitized. However, it's been theorized when you do include parasitic species in food webs, it greatly increases their complexity. And this figure on the right is showing that. So A is showing a food web that includes just free living species and basal species, where B is showing the same figure but with parasites included without concomitant predation links, where C is finally showing those predation links as well, and you can see the massive increase in complexity there. Including parasites with complex lifestyles, such as digenetic trematodes, might be especially important given the number of roles they take within an ecosystem. So in this case, on the figure on the left, I'm showing the life cycle of a trematode that has multiple intermediate hosts. So they might have a bird or a mammal that's a definitive host, but they parasitize snails and then amphibians or fish on their way to that point in their life. When they parasitize snails, they form redia, which actively produce Cercariae, which is a free-living transmissive phase of the trematode. And they erupt in massive quantities from the snail tissue. And on the right, I have a graph showing how much trematode biomass exists in some ponds. In this case, in 3 California ponds, the biomass was greater than many general taxa that you find, such as amphipods and ostracods and Ephemeroptera. And in some ponds, it's been likened to the same mass as an African elephant existing within that pond. When the free-living Cercariae erupt en masse from the snail host, they only have about 24 hours of life to actually make it to that next phase where they infect their second intermediate host. And most of them that we know will not do this. So with this massive biomass being produced, knowing that most of them won't end up actually parasitizing their next target host, it means they're ending up elsewhere within the environment. And what I'm showing in the middle here is just infectivity over time. So as these age throughout the day, it's less and less likely they'll actually successfully infect that next host, which means that they either might be consumed by predators or just naturally die off and float down into the benthos. In terms of looking at Cercariae as prey, there have been several studies that have shown there are taxa that will consume Cercaria when presented with them. However, many studies that look at Cercariae as prey do so from a perspective of how it reduces the transmission of infection from one stage to another, rather than Cercariae as actually being consumed as a food item. There have been a few studies now, though, that show that dragonfly larvae, for example, will preferentially consume Cercariae over other prey items that we consider, such as Daphnia. When considering the size of Cercariae, they fit well within the range of many planktonic taxa, including Daphnia, and I've included a comparison of Echinostoma Cercariae versus the Daphnia magna in the upper right. And because of this, it means Cercariae are far too small to directly observe in the environment to determine their fate. And thus we need to figure out a way to trace them through the ecosystem. Some previous studies have achieved this with limited success by using radioisotopes. However, these come with their own challenges and hurdles in terms of using en masse in the environment. And thus, stable isotope analysis provided a relatively cost-effective and efficient method of tracing these organisms through the environment. And this brings me to my hypothesis. I hypothesize that within a model ecosystem, cercariae-derived carbon will appear in predators known to consume them when traced with a stable isotope, in this case carbon-13. To set about testing my hypothesis, I first had to figure out how to get carbon-13 into the Cercariae to trace them through the environment. What we came up with was spiking snail food with sodium carbonate that was spiked itself with carbon-13. And the idea here was that the snails would consume this food and incorporate the spiked carbon into their tissue. At that point, the trematode within the snail would consume the snail tissue, also incorporating that spiked carbon within itself. And then when it released Cercaria, those Cercaria would be carrying that isotopic signature, which would then finally be either consumed by a predator within my model ecosystem or end up in the benthos to be consumed by detritivores. So I've included an image on the left of my setup. I set up 14 378 liter bins that I filled with filtered water, and we applied one of 3 treatments to them. So I had 2 control treatments, which every week I would only add lab water to with nothing in it. I had 2 bins that I added unlabeled Cercaria to, and then finally I had 10 bins that I was adding those spiked Cercaria to. Within each of those mesocosms, I attempted to create a model ecosystem. And to do this, I used a selection of predators that were known to consume the Cercaria, as well as a benthic detritivore and a planktonic community. So I added to each mesocosm 3 dragonfly larvae as a benthic predator, 3 diving beetles as a pelagic predator, about a half-gram ball of oligochaete worms to act as my benthic detritivores, and then a planktonic community. It wasn't entirely homogeneous, but we counted about an average of 8,040 Daphnia adults going into each of these to seed the planktonic community. And then each week during the extent of this experiment, I would go up and add the Cercaria. So to get the Cercaria out of the snails, I would leave them under a lamp, where they, after which they would shed the Cercaria, and I would collect them. And then I would sub-aliquot this total collection of Cercaria, whether they were labeled or unlabeled, and roughly per each treatment, for labeled Cercaria, there was about 105 being added a week, whereas unlabeled Cercaria, it was about 141 per week. For my statistical analyses, I first ran 2 separate univaria ANOVAs using the generalized linear model within SPSS, one for the taxa within the mesocosms and the other for the laboratory samples. And following this, we did a 2-key LSD post hoc test. And in this case, the independent variables were the addition of Cercaria, as well as the material type in either the mesocosm or the lab samples. Following this, we performed 1 sample t-tests for each lab material or mesocosm taxa to determine if the delta carbon-13 values were less negative in either the manipulated food or the mesocosms that were manipulated for carbon-13 values. To correct for the multiple hypotheses testing, as well as the uneven nature of the experimental design, we applied a Benjamini-Hochberg test procedure with an accepted false discovery rate of 0.25. Overall, the results from this experiment showed that samples coming from the treatment that received spiked Cercaria had a less negative carbon-13 value when compared to samples coming from treatments that did not receive spiked Cercaria. Note that in this case, a less negative carbon-13 value means it had more carbon-13 isotope present than a value that was more negative. In terms of the results from the GLM, both the lab sample and mesocosm samples showed significance in labeling, as well as in either the material for the lab sample or the taxa for the mesocosm samples. And finally, there was a significance in the interaction between the label and material or taxa, depending on the lab or mesocosm GLM. In this case, that shows that there was a significant difference in delta carbon for the treatments that involved the carbon labeling. When looking at the post hoc test performed for the GLM, it showed that the snail food had a significant result in terms of labeling with carbon-13, as well as the oligochaete worms. The figure on the left is showing the mean delta carbon-13 value for all of the material or taxa included in either the lab samples or within the mesocosms. The gray bars are showing when carbon-13 spike Cercariae were added, whereas the white bars are showing when there was no carbon-13 added. In terms of the one sample t-tests, all of the material in taxa, with the exception of zooplankton, showed a significant difference in terms of having a less negative carbon-13 value in the samples that were treated with spiked carbon-13 cercarial additions. In conclusion, the results of this experiment showed that Cercaria can be effectively labeled with carbon-13 isotope and traced through a model ecosystem. As well, we showed that there is the potential that benthic detritivores could be important consumers of cercarial biomass if the Cercaria die and end up within the benthos of the ecosystem they exist in. In terms of refining this experiment for the future, it might be important first to replicate a natural cercarial density within the mesocosms themselves. It is likely that the single addition of Cercaria per week that we provided did not come anywhere close to what would be found naturally being shed by snails within the environment, with a much larger number being produced continuously than we could possibly hope to add. Second, it might be good to isolate Daphnia to create a homogeneous zooplankton population to avoid including any non-specified zooplankton species that might have been present in our non-homogeneous population. Finally, it would be good to vary both the species of trematode and the species of predators present, as trematodes themselves differ in morphology greatly between species, as well as in swim behavior and size, and predators might be more keen on one species versus another, and mixing up the predators could provide a more positive result depending on what predators are added, such as potentially adding fish or another type of macroinvertebrate. And with that, I'd like to acknowledge the Koffler Scientific Reserve and the University of Toronto for allowing me to use their field space to set up my mesocosms and run my experiment, as well as Dr. Shannon McCauley for offering advice on dragonfly larvae, and finally my lab mates Kira McKee, Jamie Nguyen, and Lucy Santos, and of course Dr. Janet Koprivnikar, my former master's thesis supervisor and co-author on this paper. Thank you.